Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The advent of the 20th century heralded an age of unprecedented scientific optimism, yet the domain of therapeutics often remained a realm of empirical guesswork and profound clinical impotence. Among the great scourges afflicting humanity, syphilis, the Great Pox, stood as a particularly grim testament to medicine's limitations. Its progression from a localized sore to a devastating systemic assault on the body, culminating in madness, paralysis, and death, was a horror for which the available treatments, primarily toxic mercury and iodides, offered more suffering than solace. Into this landscape of therapeutic nihilism stepped Paul Ehrlich, a figure of immense intellectual stature whose work would fundamentally reorient the very philosophy of healing. His 1909 publication, with his collaborator Sahachiro Hata, detailing the discovery of a cure for syphilis, was far more than the announcement of a single drug. It was the triumphant empirical validation of a revolutionary concept, the sorbicutal or magic bullet, marking the birth of chemotherapy as a rational, systematic science and forever altering the relationship between the laboratory bench and the patient's bedside. The intellectual genesis of this breakthrough lay deep within Ehrlich's decades-long immersion in the world of chemical dyes and cellular biology. His doctoral work in the 1970s on the staining of tissues revealed a foundational principle: chemical substances exhibit selective affinities. He observed how certain aniline dyes would bind exclusively to specific cellular structures, coloring a nerve cell while leaving adjacent muscle fibers untouched, or staining a bacterial cell within a tissue sample without affecting the host cells. This phenomenon of specific binding, of corpora non agunt nisi fixata, meaning agents do not work unless they are bound, became the central axiom of his scientific worldview. It suggested the tantalizing possibility of designing compounds that could be selectively toxic, binding to and destroying an invading pathogen, while leaving the host organism unharmed. This vision was further refined and given a sophisticated mechanistic framework through his work in immunology and the development of his sidechain theory. To explain the action of toxins and antitoxins, Ehrlich postulated that living cells possess specific molecular sidechains or receptors which function to bind nutrients. A toxin, he argued, could cause disease by binding to these same receptors, blocking their normal function. An antitoxin worked by possessing a stronger affinity for the toxin, neutralizing it before it could attach to the cell. This model, while later superseded in its immunological details, provided a powerful conceptual tool. It transformed the cell surface into a chemical landscape of locks and keys, suggesting that a therapeutic agent, a magic bullet, could be engineered to possess a key that fit only the locks present on a pathogen. The quest for such a molecule became Ehrlich's driving obsession. His earlier efforts, particularly with arsenic compounds against trypanosomes, the protozoa responsible for sleeping sickness, demonstrated both the promise and the peril of this approach. Ataxyl, an organic arsenical, showed therapeutic activity, but its name, meaning less toxic, proved to be a cruel misnomer, as it frequently caused blindness and other severe side effects. The central challenge crystallized achieving a compound with a high chemotherapeutic index, a term Ehrlich coined to describe the crucial ratio between the dose required for a therapeutic effect and the dose at which toxicity to the host occurs. The discovery in 1905 by Fritz Schortin and Erich Hoffmann of the causative agent of syphilis, the delicate corkscrew-shaped bacterium Treponema pallidum, provided Ehrlich with a specific, visible target. The final, indispensable component for his systematic assault on the spirit arrived in 1909 in the person of Sahachiro Hata. Hata was a brilliant and meticulous Japanese bacteriologist who had mastered the difficult technique of inducing experimental syphilis in rabbits. This provided what Ehrlich's sprawling research institute in Frankfurt had lacked: a reliable, reproducible animal model in which to systematically test the hundreds of candidate compounds being synthesized by his chief chemist, Alfred Bertheim. The collaboration between Ehrlich, the grand theorist and visionary, and Hata, the master of precise experimental execution, created a synergy that would make medical history. The process they instituted was a paradigm of industrial-scale rational drug discovery, departing from the serendipity that had characterized most previous medical breakthroughs. Ehrlich's team embarked on a painstaking program of molecular modification. Starting with the basic structure of the toxic ataxyl, they synthesized hundreds of derivatives, methodically altering functional groups, changing substituent positions, and varying the arsenic's valence state, all in a determined search for a compound that would maximize toxicity to the spirit while minimizing it for the host. Among the compounds gathering dust on the Institute's shelves was the 606th derivative in the arsenobenzene series, a substance known as arsphenamine, which had been synthesized 2 years prior and initially set aside as ineffective. Upon his arrival, Hata began the laborious task of rescreening these archived compounds with his new, robust rabbit model. His scrupulous work soon yielded a stunning result. When injected into a rabbit with a syphilitic chancre teeming with spirochetes, compound 606 produced a dramatic and rapid clearance of the pathogens. Within 24 hours, the once-abundant organisms had vanished from the lesion. It was Ehrlich's Therapia Sterilisans Magna, the great sterilizing cure that could, in theory, eliminate an infection with a single, powerful dose. The announcement of these findings, first in a 1909 publication and then presented dramatically by Ehrlich at the Congress for Internal Medicine WISPA in 1910, sent shockwaves through the global medical community. The drug, which Ehrlich commercially named Salvarsan, a portmanteau of saving arsenic, was hailed as a miracle. His institute was immediately inundated with thousands of desperate letters and telegrams from physicians around the world, begging for supplies. This overwhelming demand created a logistical and ethical crisis, forcing Ehrlich and his small team to become manufacturers and distributors, carefully vetting requests and providing detailed instructions for the drug's complex preparation and administration. The reality of Salvarsan's use in the clinic was far more complicated than the clean results in Hata's rabbits. The compound was an unstable yellow powder that had to be dissolved and neutralized in a precise, multi-step process immediately before use. The subsequent intravenous injection, a technique still unfamiliar to many practitioners, was fraught with risk. Improper preparation could lead to excruciating pain, tissue necrosis, and fatal embolisms. Even when correctly administered, Salvarsan carried the inherent dangers of arsenic, with potential side effects ranging from fevers and rashes to severe organ damage. This difficult reality fueled a backlash, as Salvarsan war erupted in medical journals, with critics highlighting treatment failures, relapses, and fatalities. Some dismissed Ehrlich's methodical approach as mere pharmacological cookery, while others, perhaps motivated by professional envy, accuse him of unseemly commercialism and reckless endangerment of patients. Ehrlich responded to these critiques with the same scientific rigor that had led to the discovery. He tirelessly collected data from clinicians, meticulously documenting over 10,000 cases to refine dosage and treatment protocols. He acknowledged the drug's limitations and dangers, working to educate the medical profession on its proper use. His commitment to improvement led directly to the development in 1912 of neoselvarsan, or compound 9-14, a derivative that was more soluble and easier to prepare, significantly improving its safety profile. While Salvarsan itself was eventually superseded, its ultimate legacy lies in the profound paradigm shift it inaugurated. The discovery of compound 606 was the definitive proof of concept for chemotherapy. It demonstrated that a deep understanding of chemistry and biology could be harnessed to design and synthesize molecules with specific therapeutic actions. This principle of rational drug discovery became the guiding light for subsequent generations of researchers. Gerhard de Mag, working at the German dye conglomerate IG Farben in the 1930s, was an explicit intellectual heir to Ehrlich. He followed the same systematic approach of screening synthesized dyes for biological activity, leading to his discovery of the antibacterial properties of Prontosol, the first of the revolutionary sulfur drugs. Even the story of penicillin, while initiated by Alexander Fleming's famous moment of serendipity, owes its therapeutic fruition to Ehrlich's model. The subsequent work by Howard Florey and Ernst Boris Chain to isolate, purify, test, and ultimately mass-produce penicillin was a monumental exercise in the very chemotherapeutic principles that Salvarsan had pioneered. The arrival of penicillin in the 1940s, a far safer and more broadly effective antibiotic, rendered usfenamine obsolete for the treatment of syphilis. The magic bullet that had vanquished the great pox was itself vanquished by a more magical one. Yet, the intellectual framework that produced Salvarsan has become more dominant than ever. The core concept of selective toxicity, of designing molecules to target a pathogen, a defective protein, or a cancerous cell, is the foundation of modern pharmacology. Every time a scientist develops a new antiviral drug that inhibits a specific viral enzyme, or creates a monoclonal antibody that targets a unique receptor on a tumor cell, they are walking in Paul Ehrlich's footsteps. The development of targeted cancer therapies like imatinib, which precisely blocks the action of the rogue enzyme driving chronic myeloid leukemia, is a magnificent modern realization of the side chain theory and the magic bullet dream. The 1909 collaboration between Paul Ehrlich and Sahachiro Hata was a watershed moment in the history of medicine. It was the point at which drug discovery transformed from an art of folk or unfortunate accident into a science of deliberate design and systematic investigation. The conquest of syphilis by Salvarsan was a monumental clinical achievement, but the enduring triumph was the establishment of a new way of thinking. It validated a powerful idea that through rational inquiry and chemical ingenuity, humanity could craft weapons of exquisite precision to combat its microscopic foes. The century-long quest for ever more specific, more effective magic bullets, a quest that continues in laboratories across the globe, remains the most profound and lasting legacy of a historic breakthrough.
We really appreciate you tuning in to this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.